develop, the smarter that the bacteria get, so to speak, and the better that they become resistant to it. Some bacteria are born resistant to a particular antibiotic, and so we call that innate or intrinsic because they're born with it in them. Acquired means they got it from one of those routes that we talked about in the genetics chapters, so horizontal gene transfer, right, or spontaneous mutation. So transduction, transformation, or conjugation would be all the types of horizontal gene transfer, all the ways that your friend or your neighbor could give you, if you were an E. coli, could give you the resistance genes that they have. So mechanisms of action of antibacterial drugs, I just wanted to reiterate how important it is to realize that the goal of an antibacterial drug is going to be what? To kill the bacteria and not kill you, right? And specifically, if we can, we'd like to kill the particular bacteria that is causing your issues and not kill the other species that you might have living in you too that are doing you a favor and living in peace. So that's why there's stars by cell wall synthesis, because the biggest and best prescribed class in this section is the cell wall synthesizers. And that should make sense to you. Why? Because protein synthesis is certainly something that you can target for an antibacterial, nucleic acid synthesis, cell membranes, metabolic pathways. But all of these things, for the most part, have a lot in common with our cells, right? They make proteins in the ribosomes using RNA. We make proteins in our ribosomes using RNA. They make DNA from DNA. We make DNA from DNA. So all of these things are very similar, whereas what is the difference between their cell wall and ours? Well, we don't have one, right? Our cells don't have a cell wall, and theirs do. So that would be the most obvious thing to target. That would be the most different from us to them. And sure enough, there's a whole bunch that fall into this category, and it's a lot of the ones that I'm sure you've heard of before. So I'm not going to read all of the stuff when it comes to the individual drugs. I've already suggested and will continue to suggest that this is a very good thing for flashcards. But I will say, for one example, I'll take this slide because we're on it now, what I would write on my flashcards if I were making them. So beta-lactam drugs would be on the front of my note card for this one, and on the back it would say penicillins, cephalosporins, carbapenems, and monobactams. It would say antibacterial drugs that inhibit cell wall synthesis. It would also say only effective against actively growing cells. And that's important, but that should make sense to you. Why? Because if you're inhibiting cell wall synthesis, when would you be synthesizing a new cell wall, right? When you're growing or you're reproducing. So this is unfortunately true for a lot of the the drug classes, that it's only effective against actively growing cells. Interferes with peptidoglycan synthesis, can be broad, which we already said covers a lot of different kinds of bacteria, or extended spectrum, which is actually like even bigger, so that kills pretty much anything. It's a very, very effective class of drugs. So these are all things that I would write about that on my note card. When you get down to like bacitracin, I would definitely write that, you know, it's an antibacterial drug that inhibits cell wall synthesis, and I would also write toxicity limits it to topical applications. Vancomycin, definitely antibacterial drugs that inhibit cell wall synthesis. Poorly absorbed, PO means by mouth, so it's given IV. That's in bold, it's underlined, it's definitely important. Important for resistance, often last resort, resistance is increasing. Those are all things that I would include, but I would focus more on the more important things. Alright, so this is more stuff to go on note cards. It should make sense to you that inhibiting protein synthesis would be static, not cidal, because you're inhibiting their ability to grow and reproduce, surely, when you're inhibiting their ability to make certain proteins, but it's not going to straight up kill them. And it's important to note that when we talk about drugs that inhibit protein synthesis, how are they doing that, right? We said that we do protein synthesis very similarly to what they do. We use mRNA that's made from DNA that goes out to the cytosol and gets made in the ribosomes. So the only thing we can do is sort of target the difference between their ribosomes and ours, which lies in the size. So theirs are something called 70S, and S is just a way that they measure size of ribosomes. And theirs are 70 and ours are 80. But as you can see, there's going to be less of these than the cell wall ones, and they're going to have a greater chance of side effects because there is going to be some definite overlap from their ribosomes to our ribosomes. Alright, so there's some more note cards, stuff that's good for note cards. Write everything that's on here on note cards for sure. This is a good note card as well, but I brought up the fact that there are only a few that interfere with metabolic pathways. Why? Because as much as we look at bacteria and we look at us and we want to see all the differences between them, at the end of the day, their DNA is very similar to ours, and all living things, all living cells, have very, very similar processes as far as they eat, they sleep, they poop, and so their metabolic pathways are not very dissimilar from ours. So the more drugs that we developed in this class, the more horrible the side effects would be from them, because the more overlap they would have with affecting our cells. The one exception is folate inhibitors, and as any woman out there who's had a baby knows, you have to take folic acid when you're pregnant, and that is because why? Because we don't produce it. We have to eat it, whereas bacteria can produce it. So if you can target that pathway in particular, that would be useful, right? Because it's, again, a big difference between them and us, and the sulfa drugs are the ones that do that. You can interfere with different types of things that are in their cell membranes that aren't ours, but some are limited to topical applications because they have nasty side effects. So Neosporin is very effective topically, but it's not something you'd want to eat for sure. You'll notice that up until this point there have been some drugs that only affect gram-positive or gram-negative, some that only affect anaerobes, but we haven't talked about a particular genus or species of bacteria all alone and the way to treat it. The only one that we'll do that with is Mycobacterium, and that's because these guys are really a big health concern. So if you remember, the two of the major ones that fall into this category are tuberculosis and leprosy. So they're a giant problem. And the other reason is because they're very, very difficult to treat. So that's twofold, the reason for that. One is they have this really strange cell wall, and it's thick and it's waxy, and it has these mycolic acid things that normally are found in fungus, not in bacteria. And that prevents the entry of a lot of drugs, and so I'm sure you can imagine that if the drugs can't get into the bacteria, they're not going to be effective against killing it or slowing it down. The other problem is that growth is slow. So like we said before, a lot of the drugs only work on growing or dividing cells. So if they're not growing or dividing, even if you manage to get the drugs in a very slow fashion, it's going to be a long time before they work, and the chances are they'll be metabolized and getting rid of before then. So it's a very harsh treatment that we use for tuberculosis and other Mycobacterium species. The group of 5 that are preferred are called first line, and they're given, first of all, they're given in combination, and second of all, they are given for a minimum of 18 to 24 months. So that's why I've said this before in class, you do not want to be exposed to TB if you do not have to be ever, because if you are, you will be on nasty combinations of drugs for a minimum of 2 years. There's one in particular that's used a lot, isoniazid, as part of that first line combination, but unfortunately some resistance has developed, those multi-drug resistant tuberculosis strains that we talked about in emerging diseases chapter. And so second line drugs can be used for resistant strains, but they are less effective and or have a greater toxicity risk. So if you remember back to the, I'm not going to scroll there, but if you remember back to that slide where we're looking at the therapeutic index, that top number, the 2,500 milligrams, corresponded to what? It corresponded to the amount that it would take to make you sick, right? And unfortunately, the only way that we can figure that number out is by testing in humans. So the bottom number, the 500 milligrams, can be tested in lab, thankfully. And anytime that we can test things in lab, as opposed to making people sick just to get the results, obviously is preferable. But I do put this disclaimer on there: inhibition does not necessarily mean successful treatment. So that's just kind of a way of saying that, you know, just because this is what we found in lab doesn't mean necessarily that that's what the case is. But basically, these are the two things that we calculate to try and find that bottom number. And the difference between using an MIC and an MBC comes from the fact that MIC, minimum inhibitory concentration, is the lowest concentration that prevents growth in vitro, which is in lab. And this has to do with what kind of drugs. What type of drugs prevent growth? Bacteriostatic drugs, right? Whereas minimum bactericidal concentration, or MBC, is the lowest percent that kills. So this is going to be the test that we use to determine that bottom number if the drug is bactericidal, and this is going to be the test that we use to determine it if the drug is bacteriostatic. So I'm not going to read this one. You can read it, and I have been told that you are doing this or have done this already in labs, so you should be pretty familiar with it. If not, I'm sure you can find a good YouTube video or Google it and read about it. This I just wanted to talk a little bit about. We keep saying resistance is a problem, there's all this resistance going on. And the question is, so you get this gene that allows you to be resistant to drugs. What exactly does that mean? Well, there's a lot of ways that you can be resistant to drugs, right? You can have drug inactivating enzymes. So what does that mean? That means that you have ways of basically chewing up the drug before it harms you. So you're an E. coli, and this person that you're living in takes a drug like penicillin, and the drug comes into your cell, you take it up, and before you can get to the point where you're going to die from it, you have these special enzymes that you can make that just destroy the drug. Alteration in target molecule. So the target molecule is what the drug is targeting. So in the case of like protein synthesis inhibitors, for example, they target what? Ribosomes, right? So if you can mutate your ribosome in some way, alter your ribosome in some way that the drug can't affect it anymore, then that would make you resistant to that drug, right? Decreased uptake, increased elimination. Basically, you have to get the drug into the bacteria for it to work, right? So if you can prevent it from getting in or push it back out as soon as it gets in, that would make you resistant. So in the chapter on horizontal gene transfer, we talked about the plasmid, the little piece of DNA that holds the conjugation plasmid. So the one for the bacterial sex was called F plasmid. So big surprise here, the one that holds the resistance genes is called R plasmid. I'm not going to read this, but it's unbelievably important, so I hope that you will take the time to read it. It's pretty self-explanatory, and like I said, it's very important, and not, yeah, this one as well. Alright, so antiviral drugs are different. They have to work differently than antibacterial drugs, and they're harder to use and harder to develop, and they usually have much nastier side effects. And the reason just has to do with what is it that you're treating. And so in the case of an antibiotic, an antibacterial, you're treating a bacteria, which is a cell, right? So if you can target something like a cell wall, which makes their cells different than our cells, then you're killing specifically their cells for the most part and not ours. Whereas in the case of viruses are non-living, right? And so if you have a virus infection, if your patient that you're treating with these drugs has a virus infection, what does that mean? It means that there are viruses inside the patient's actual cells, whether it's their lymph node cells or their blood cells. It's somewhere in their body. and inside of their cells. And so the trick is